Hello everyone. Today I will present my work on the genetic basis of coat color variation in long-tailed weasel. Coat color molt from summer brown to winter white is a crucial phenological adaptation of over 20 mammal and bird species. It's triggered by the photoperiod and it's essential for these animals to maintain camouflage year-round in the habitat seasonally covered with snow. However, global warming is disrupting this adaptation, and even in different scenarios of future climate change, it's expected that snow cover extent will reduce in the next century. And this is causing a color mismatch between white individuals and the background, with severe consequences in their survival rate due to increased predation. And so understand how this coat color polymorphisms evolves and the pathways of the evolution of this coat color polymorphism is essential to understand how these species will cope with future climate change. As some of these species, there are individuals that remain brown year-round, while others change their coat color to white during winter. So in this study, our aim was to unravel the genetic basis of winter coat color polymorphism using museum specimens. And as a model study, we used the long-tailed weasel, which is a species widely distributed in North and Central America, and it's polymorphic for the winter coat color. In this map, you can see that dark and black colors mean higher probability of winter brown phenotype, while light gray colors mean higher probability of winter white phenotype. And so in North America, there is a very large region of transition between coat color morphs, extending from the East Coast to the West Coast. I think the advantage of this large transition region and of the excellent records in the natural history collections, we sampled 98 specimens for two transition regions, one in the East Coast and another in the West Coast, and produce whole genome and target capture data for these specimens. Whole genome low coverage data was produced for 44 specimens, 11 specimens for each color morph per region. And the first assessment on the population structure using a PCA analysis and ancestral clustering analysis showed a clear differentiation between the regions, meaning that geography rather than coat color are the main factor for population structure. And so we have two independent regions of the transition region between coat color morphs. So using these two transition regions, we produce whole genome scans, and with window-based FST scans showed a single common region of association to the winter color morph, a region of 300 kbs at the beginning of a scaffold. Additionally, taking advantage of these replicated transition zones, we produce case control association scans, and we use a CMH test that looks for similar allele frequencies changes in coat color morphs. And the same regions pops out, a region that we look in the annotated genome of the ferret. And it included several genes, including the MC1R gene, which is a pigmentation gene and a transmembrane receptor that, in absence of the protein ZIP, the alpha MSH induces the MC1R activity, leading to the production of the dark brown pigment, while in the presence of a ZIP, this protein works as an antagonist of the MC1R activity, leading to the production of pheomelanin, the yellow and red pigment, leading to lighter coat colors. So we further investigated this association region by producing target capture data for 85 specimens. And PCA and clustering analysis showed a clear differentiation between white and brown phenotypes. We further explore this region by analyzing the topology variation across the association, the target capture association region. And here you can see on the left the association region, and the dominant topology was a topology clustering together both brown phenotypes. It's the green topology, meaning that the brown phenotype had a similar evolutionary origin in both regions, while outside the association region on the left, the predominant topology was the one correlated with geography. So we then Sanger sequencing the MC1R gene, and we found that the white samples were homozygous for an allele, for the complete and ancestral allele. Here you can see a 2D model of the MC1R protein. However, for brown samples, we found alleles with deletions, and they were homozygous or heterozygous for these deletions. In the Western region, we found 2 deletions, one in the second transmembrane domain and another in the first extracellular loop. While in the Eastern region, we found a single deletion in the second transmembrane domain. While we do not have some functional analysis on this, on the impact of these deletions, previous results for deletions here in blue and green, and mutations in red, showed that these mutations and deletions cause darker or a brown phenotype. And this was caused by a constitutive activation of the MC1R, leading to continuous production of the eumelanin, the dark and brown pigment. So in this work, we showed you that a genomic region that surrounding the MC1R is associated with the winter coat color polymorphism in long-tailed weasel. Additionally, we showed that the deletions associated with the brown winter phenotype should have a similar common evolutionary origin, and that these deletions are likely causing alterations in protein function, leading to the winter brown phenotype. Just to finish, I would like to thank my research group at CBU in Portugal and to the funding agencies. Thank you.